Welcome to the University of Pittsburgh Health and Explainable AI podcast, a collaborative initiative between the Health and Explainable AI HexAI Research Laboratory and the Department of Health Information Management at the School of Health and Rehabilitation Sciences, and the Computational Pathology and AI Center of Excellence, CPACE, at the University of Pittsburgh School of Medicine. I'm your host, Jordan Goss-Poire, a health and science reporter. In this podcast series, we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine. From reshaping diagnostic accuracy to enhancing patient care pathways, we'll highlight how AI is creating new bridges between researchers, clinicians, and healthcare innovators. Led by Ahmad P. Tafti, Houman Rashidi, and Liron Pantanowitz, the HexAI podcast is committed to democratizing knowledge around ethical, explainable, and clinically relevant AI. Through insightful conversations with domain experts, AI practitioners, and students, we'll spotlight the latest breakthroughs, discuss real-world applications, and unpack the challenges and opportunities that lie ahead in responsible AI in healthcare. So, whether you're a student, practitioner, researcher, or policymaker, this is your gateway to the future of AI-powered healthcare. Today, on this podcast episode, we're going to speak with Daniel Heller, head of the Cancer Nanomedicine Laboratory at Memorial Sloan Kettering Cancer Center and Weill Cornell Medical College. Daniel's laboratory develops nanotechnologies for the research, diagnosis, and treatment of cancer and allied diseases. We're going to discuss nanotechnologies and nanosensors applied to advanced diagnostics. And we're going to discuss explainable AI in detail, applied to Daniel's work. Daniel, welcome to Pitt's HexAI podcast. To start us off, would you like to introduce yourself and tell us what first sparked your interest in nanotechnology?
Thank you so much, and super great to be here. So I got excited about nanotechnologies in my freshman year of college when I was at Rice University, and that was the year that the Nobel Prize came out for two professors at Rice University who got the Nobel Prize for the buckyball, the carbon-60 molecule. And carbon-60 was just this, basically a soccer ball of carbon. But if you extend it into a tube, the carbon-60 becomes basically a carbon nanotube. And that's what I study in order to develop sensors for cancer.
It's nice to hear that all the interest in carbon nanotubes sparked practical research like yours. Could you walk us through nanosensing technology and explain what makes working at the nanoscale so unique and exciting to you?
So there's really an explosion of different methods and technologies to get information out of the body and into potentially a computer, or at least to a wearable device, to a computer, to be able to then use that data and process that data with now such increasingly complex and interesting methods like different forms of AI. And we found that when working on carbon nanotubes, that they really work as a potentially unique form of sensor technology that can transduce information. And so certainly there are wearable technologies that shine light on the body and use the reflection of that light to measure things like heart rate. And what we want to do is use nanotechnologies, which are, super tiny materials like nanoparticles or carbon nanotubes. They're so tiny, they get down to about 100,000th of the diameter of a human hair in terms of that size range, and then use those to pick up information from inside the body or even just in blood and get that information to either a wearable if it's part of a device that could be worn, or just into a computer. And that information could be very complex or very simple. It could be, say, one biomarker, one protein level, or it could be many, many, many events that can be used to understand the state of the body, the state of a disease, whether someone has cancer.
For students specifically interested in microsensing technologies and their incorporation into healthcare, can you elaborate more on the field to help students visualize the range of different subfields and applications that researchers are advancing, and tell us more about your area of research and what the cutting edge of nanosensing looks like to you.
So there's many types of technologies here that kind of fall under these sensor and nanosensor and also kind of nano robot or micro robot types of fields. And so when kind of coming down from the larger to the smaller, there's many micro technologies that people are working on, which are definitely larger than the things that I work on. There, they might be these pill-sized robots with microprocessors inside of them that are basically like small computer chips. And so a pill size, say, for instance, a pill size camera that someone wants to ingest in order to do kind of a colonoscopy without having an actual endoscope where people, and this has been done and realized, and or pill size sensors for there's been pill size sensors that are kind of similar to these cameras but maybe can sense different chemicals. So those are definitely larger than the things that I work on, but still they're miniaturization of electronics into something that can be put inside the body. And certainly things they're also related to things like glucose, monitors that you can put on the body, and these kind of implantable and some wearable sensors. And these can be very made to be very smart. You could put maybe almost a whole computer chip onto these things, and they could potentially in the future be used to do things like people want to do, robotic surgery using, micro robots and things like that. And those are definitely doable. And they can be made very smart because you can put a whole computer on these things. And I would say that they, maybe a competing size and similar types of size technologies are things like even engineering bacteria. So, and engineering where you can make bacteria sized and cell sized materials very smart. They can have a nucleus. They can have a lot of instructions in them. You can do so many smart things. Whereas if you get down to nano size materials, they are exciting because of their potentially because of the properties, the electronic or optical properties that they can have at that scale. Like, for instance, why I work on carbon nanotubes, these tiny tubes that are only 1 nanometer, more or less, in diameter. So that's what's about 100,000 times smaller than the human hair. And at that size, we look at them because they glow and they fluoresce in the infrared wavelengths. And those wavelengths are exciting to us because the light can pass through tissue, and we can make, say, an implantable device that can glow, but it give off that glow, the color that they glow at can actually get through tissue, and we can make something that can transduce, or get a signal from inside the body to outside the body using light. And so we really like those materials because of these properties. But it's not really smart in the way that a robot or a bacteria can be made smart. It can't make decisions. It's super tiny. And so that's why I always think about when people kind of, I guess maybe 20 years ago, when people were there was a little bit of a scare of nano robots taking over the world, and people working in nanotechnologies were looking at these things and saying, like, no, these are not going to be that smart. Something at that size can't even carry all the instructions to be smart. They can have cool properties, but they can't be smart in the way that something larger can be. So what I work on is these tiny nano sensors that can pass light out from inside the body, for instance, and transduce an event. And in this case, it might be a binding event of a biomarker, where we can take a biomarker that's inside the body. We want to know, let's say, a protein level inside the body that can tell someone something about disease, and we want to get that information out of the body. And so we think about putting these sensors inside the body, maybe right under the skin. The light can't go that far through the body, this infrared light, but we can shoot light at the body, get this infrared light signal outside the body, and we can measure that even maybe with a wearable device, and then get kind of real-time information. Now, where AI comes in is that, and really the things I've been doing now with these sensors, is that I started really comes from when I first started doing work at my institution, Memorial Sloan Kettering Cancer Center, and started asking, what are the big problems we can solve? And really the things that I wanted to solve with my technologies, the reason why I came to this institution, came to a cancer center with a kind of an engineering background and chemistry background, I wanted to do two things. I wanted to treat metastatic cancers and detect cancer early before it becomes metastatic, because metastatic cancers are some of the most deadliest form of the cancers and really the cause of most cancer deaths. And so we thought, well, how do we detect cancer early? So I started in the lobby of the building that I work in, where I am now. I ran into a gynecologic oncologist, an ovarian cancer physician and researcher. And he said, we really need to detect ovarian cancer early. And so it would be great to have a sensor that can detect ovarian cancer. And I thought, great, this is exactly the kind of problem that we need to work on, because it turns out that ovarian cancer is very treatable when it's at stage 1 cancer, but it's very hard to treat at stage 3 or 4. And so if we could just detect cancer early, at stage 1 or even at stage 2, or certainly as much as possible early as possible before stage 4, we can make people live longer and maybe even cure ovarian cancer in patients where we can detect it early. So we said, great, let's do this. And what do we do? Okay, so we have a sensor technology. We could potentially make it detect anything we want. Which we detect? And so we started trying to detect the known biomarkers for ovarian cancer. And the engineer really said, like in my engineering mind, I said, okay, I have the biology and the problem. So let's go and try to make a sensor that works better against ovarian cancer and detect these markers better. And we started doing that. But eventually we realized that the problem is that the markers we're detecting are just not good enough. That it's not that we can't detect that marker better. We want to detect a more sensitive a sensor for those markers. It's just that those markers just don't appear at the right times in early stage ovarian cancer. They appear too late, or they appear they're at high levels in patients that don't have ovarian cancer because of some other condition. So they're just not specific enough. There's really no one marker that anybody's found for ovarian cancer that's specific enough where you can just take a test and see and look for that one marker. And so we wondered, well, we have a sensor technology that we could potentially make to detect anything. These carbon nanotubes, they glow in this infrared color. But the other thing they do is that they're basically all surface. If you think of a tube, there's nothing inside, at least inside of a carbon nanotube. And the way that they glow is that there's the electrons on the surface of the nanotube, basically, that are related to those carbon atoms, and they're all carbon atoms. There's nothing else that the carbon nanotube is made out of. It's just these hexagons of carbon, which is basically graphene or graphite, but rolled into a single tube. And those atoms have the electrons on these atoms are enabling them to glow. Basically, they're getting, if sometimes physical chemists say, well, they get to an excited state and they drop down to a lower state, or material scientists sometimes say that there's an electron in a hole, which is a positive area, and they recombine and they give off light. That's just kind of the way that people describe why things fluoresce, give off, glow, like black light, objects that fluoresce in UV light, for instance. These things, we can actually shoot red light at them, for instance, and they give off this infrared glow. But they're very sensitive to anything near the surface, because something that sticks to the surface can perturb those electrons on this that are causing the glow, the fluorescence, and it can change the intensity of this glow, or the wavelength, the color, just slightly. And so each nanotube can give off this color, this glow, and we can monitor the intensity and wavelength of it. And we've been able over time, over the last, say, 24 years since it was discovered that these things can glow, actually, that of all the reasons that they can be perturbed, that this fluorescence can be changed in intensity and color. And people have made different types of sensors for specific things. But we said, well, we don't know what we want to detect with these sensors. We know that there's must be lots of markers in the blood that show up, and at some point in a cancer patient, they're going to change. And the question is, can we see these markers change enough in cancer patients that we can figure out, that can see a signal difference with these glowing of these nanotubes when things stick to them, that will differentiate kind of the signals between cancer and normal. And what I'm really talking about here is not one sensor now. We're talking about making a bunch of sensors, an array of sensors, because we don't know really what's going to give us basically what we call a fingerprint of cancer, of disease. So what we do is we make something that resembles the way your nose works. And the idea is now we're making a sensor technology based, instead of on recognition of one marker, we want to make something that can give a sensor the mechanism of perception, or the power of perception. So what is perception? Well, your nose works because you have many sensors, basically olfactory receptors in your nose, in your sinuses, but not that many, about 400. And it works if you smell a cookie, and you can all picture the smell of cookies, a chocolate chip cookie. So the question is there a chocolate chip cookie molecule? And the answer is no. There is no one molecule that's chocolate chip cookie. But there are a bunch of different molecules that come from chocolate chip cookies. And when you smell them, they bind to these olfactory receptors, some of these 400 different receptors, so these sensors, and it creates a pattern, a bunch of different molecules from the cookie, a bunch of different sensors in your nose, and that creates a fingerprint, a pattern. And that pattern goes to your brain, and the brain recognizes it as cookie. So we wondered, can we use this type of analogy, this type of technology, where we have many sensors, they all kind of stick to different things. We don't actually know what they're, a priori, or originally we don't know what they're sticking to. But if we can make a bunch of different sensors that stick to a bunch of different things, in this case, from the blood, can we make a fingerprint instead of your brain, that AI could recognize? And so what we do is we take a bunch of our different nanosensors, and we basically can take our nanotubes and we can modify them differently. We can change the surface so certain things can stick to them and certain other things can't. And we can put little molecules on them that change the sensitivity of them to things like pH and charge and hydrophobicity and things like that. And so we have basically modified our carbon nanotubes to all be sensitive to different things, so that when we put them into the blood, and in this case, we're taking the blood out of the body for now, not putting them in the body, just this type of sensor in the body just yet. And all the molecules in the blood, these proteins, can stick to the different nanosensors and cause changes in each of the sensors' intensity and wavelength, color and wave and intensity. And then we basically can get a lot of data, lots of different nanosensors with different intensities and color changes. And so we have basically a fingerprint, a bunch of different signals. And those signals we can take from, say, a bunch of healthy patients, and blood from a bunch of healthy patients, and blood from a bunch of cancer patients, and try to see if those signals are different. And so that's called a supervised machine learning model, because we're telling the machine learning model, okay, these are the cancers and these are the non-cancers. Here's all the data from all these different signal changes of all these binding events from our nanosensors to the different molecules in the blood. Can it tell a difference? And we ask it that. And so what we found on the first try with just a few hundred patient samples is that it could do already better in differentiating cancer and normal than known cancer biomarkers, than the approved cancer biomarkers. And so we were really excited about that. And it was kind of like a Hail Mary pass. We're like, why is this thing, how could we make a sensor where we just don't know why any of our sensors are going to work? And so then we got this really cool data, and we validated it and really made sure that it was working. And then we did it again for another disease. We did it for brain tumors. Can we differentiate brain tumor versus healthy? And we even tried to ask, could we differentiate different brain tumor diagnoses from each other, like glioblastoma from meningioma? And it turns out it could do that fairly well, but we didn't have as many samples then, so it wasn't doing it as well as tumor versus normal, where we had lots of tumors of all different brain tumors, could differentiate really, really well. Definitely better than anything we've seen. And so we're really excited about that. And that paper is coming out very soon, by the way, in a journal. And so we realized that we are starting to believe this now, that we're really able to separate out blood from patients by disease or by different questions that we have. And it doesn't need to be just from cancer, because it's not using anything specific from cancer. it's using proteins and maybe small molecules that are binding to the sensors. And then, but still, we wanted to know that. But what is actually causing these things to work? What is the mechanism? And so, what is sticking to these things, to these sensors? And so we first looked at proteins because there's a lot of machines, mass spectrometers that could measure proteins. And we started washing off the best sensors, the sensors that machine learning feature importance analysis was telling us, which are the most important sensors. And when we washed it off, we were seeing all these proteins that are relatively rare in the body. So it was kind of de-enriching the most common proteins like albumin and stuff that you really want to get rid of if you want to see lots of good signals in your mass spec. And we're finding all these rare proteins, and the ones that were showing up the most, for instance, in the brain tumor cases, some of them were known cancer biomarkers in brain tumors. None of them very specific enough to say like it's a marker that everybody would want to look at to know if someone had brain cancer, but studied before. And so that was telling us, oh, this is actually potentially enriching these biomarkers for cancer and enabling us to potentially discover new biomarkers. And if we found those old known biomarkers, and when we found new ones that we started looking at and trying to validate by going back to more patient samples and saying, oh, these actual enriched, are they turning up at higher levels in brain tumor patients? And they were. So it really was interesting because for us, we wanted to know just cancer and normal and decisions of that treatment, but then we've ended up finding out a new way to discover biomarkers where the sensors that were participating the most in the decision making and that we used AI for to process the AI process was telling us which of the sensors was most important and potentially sticking to the most biomarkers that were potentially showing up at higher levels or changing the most in the disease.
What kinds of students would make a good fit to work in your lab?
My lab looks for, especially on these types of projects, looks for people who we know we're never going to find someone with exactly all the skills. And we train people who have some programming skills and can apply them or can do some of the AI work, but also people willing to really go into ask patient related questions and disease related questions and kind of collaborate a lot because we have the nano sensors that we have to make and we do some chemistry and we to develop these. So we have some chemistry, material science, some real patient clinical questions to dive into and understand the biology of this as well as the programming and AI. So it's kind of a renaissance people that really, really like this and kind of get into it and can put all these different parts together.
On this podcast, we like to ask all of our guests to share for us how they view explainable AI and explainability. And also, we like to ask our guests to offer students a research project idea. So, combining the questions together, what's something that you'd love to see students tackle, incorporating explainability? And to throw in 2 more challenges, how can universities like Pitt prepare students to work in labs like yours? And how can we collaborate on an institutional level?
So certainly explainability has been a tall order in the world of AI. And so I'm luckily in the area of machine learning, which is maybe a little bit less black boxy than large language models. I think that in part because the data you put into it is pretty much, in our case at least, the data that we're taking in our lab, it's that we have some control over, that we just have a lot of it, and it's hard to wrap our minds around all the different things that we put into that model because there's just so much of it. But it's all, in our case, nano sensor data, sensor interactions, of carbon nanotubes binding to our quantum whale nanosensors that we developed, binding to proteins. And so we have all these protein nanotube interactions. And what we do is then make this model out of those interactions and use that to diagnose disease. So in this case, we have lots of things dials to turn, things we can ask, things that students could do and that some of them are doing in our lab. One big area that we've been working on is certainly figuring out, our main question has been, what is actually causing this response? What are the actual proteins that are binding to our sensors? And so after doing the machine learning, and what we can do is that now go into the nuts and bolts of machine learning, and we do what we call feature importance analyses and figure out what features are causing the response, the most of the response, or as much as possible, the most important features in causing the response. But there's other things that we haven't done also, which is SHAP analysis, and there's something called LIME, which are also ways to make these models more explainable and figure out what features are important. And so we've done some of those, we haven't done all of those. We definitely want to do more. But what the feature importance analysis has allowed us to do is to figure out which sensors are most important. And then what we've been doing is then washing off those sensors that have been sticking to all the proteins in blood, plasma, and figuring out what is stuck. And the question for us has been, what is sticking to those sensors? And are the proteins sticking to the sensors actually causing the response? And can we discover new things from those, what's sticking? And so we have been washing off the proteins and doing proteomics, mass spectrometry, working with people with the big, mass spec machines that can really tell you a lot now about what proteins you have. And what we found is that the most important most important sensors that we wash off have been telling us really an interesting biological information about what's sticking to the sensor. And what we found first is that some of those proteins that we washed off were known for our brain tumor detection project. Some of those proteins are actually known in the glioblastoma literature. They're discovered biomarkers. And so we're really excited about that, hey, we first figured it was a good gut check to say, well, there are people that have in this field of proteomics that didn't use our methods, but used their own methods and found some of the same glioblastoma biomarkers. So great, we know that at least we're not going crazy here. And then what we found is that this enrichment onto our nanosensors has really washed off or prevented the binding of a lot of really common proteins that kind of block a lot of mass spec findings, where the albumin and things that are really common in the blood don't really stick as well to our sensor. So then we found that it enriches a lot of rare proteins. And that's really exciting, because when you can enrich the rare proteins, those are the ones that are most likely to be changing in the context of disease. And we found that a lot more different proteins are sticking to our sensor than we can normally find in just kind of like unpurified blood. And the nice thing about this has been that we know that a lot of those enriched proteins are the ones sticking to the sensor that cause a lot of this sensor change that allow us to do the diagnosis. And so we've been diving into this, and now we have a biological question. What's going on here with these proteins? Which ones are important in these cancers, and where do these proteins come from? And what we discovered originally with brain tumors is that some of these proteins are from the brain tumor themselves, but others are from different parts of the body, like the environment of the tumor, but also the whole immune system. And so what we've been excited about is this idea of the tumor ecosystem, that tumors are not just the tumor cells themselves, but there's the microenvironment, which people have been, around the tumor, we've been interested in, but also the whole body responds in different ways. And there are cytokines and different molecules floating around the blood. And we're looking at the blood that you take from the arm to diagnose brain tumors. So we know that whatever could be changing in the blood is important to us. And we're now diving in to understand what's going on in the whole tumor ecosystem of a brain tumor patient. And one could definitely look at these different proteins and do a lot of work literature-wise and computationally to understand the findings from just these proteins. So now even just looking at the data that we got, we just hit the surface of this and haven't gone very deep yet, because we wanted to understand this method and can we diagnose with our sensors. And so there's just a lot to do there. And so this is kind of really based on my own, research and the paper that we just published. But I know that there's just so much more that we could learn from it, and also other ways that we could potentially improve it by different analysis methods. And that's even before going in and tweaking with our sensors and making more sensors, which we definitely can do, and seeing if we could understand which sensors to make next for a new type of disease. What we'd love to get to is like a universal sensor set that could be used for any different types of diseases, and then and all we have to do is take blood from different patients. So there's a lot of projects, a lot of directions that we're thinking about.
On a data sharing level, where do you see collaboration opportunities?
So luckily for a lot of these proteomics findings, we get tons of data that we put online, we put onto in our papers, but also onto repositories. And so that kind of allows us to kind of immediately collaborate with people who want to do work on the tumor ecosystem and discovering new biomarkers. And so talking to biologists and clinicians, and this is important, and people who are just love to dive into data, and students who would love to get involved in this. This is definitely a great way where we produce this data and now have a huge data set that just needs to be dived into to really understand more. So I think that's kind of an easy, fast way to get involved in a research project related to this. And then certainly we realize that there's people that are on the computational side that are making new algorithms. What we really want to understand is how to deal with sparse data sets, because our data always starts with patient samples like blood. And there's just not that many of them for certain diseases. We're talking about brain tumors that are rare diseases. In the past, we're also doing work on ovarian cancer, which is a rare disease. And one big question on the machine learning side is, what do it do when you have not so many samples? And what are the best algorithms for that? And can one make better ones? And so just on the computational side, on the machine learning side, how do you deal with sparse data sets? And, I know that nobody can make magic out of nothing, but when we get down to fewer samples, and we love to get hundreds or thousands, but sometimes we don't even get hundreds. And what we find is that when we get down below 100, we don't usually have enough data to make a great model. And so people making better machine learning models for those sparse data sets is very valuable.
All right, thinking about the future, how would you love to see labs, hospitals, and academic institutions collaborate more closely? To close out our interview, what would be your futuristic wish?
Well, for just the research side, my wish would be to find better ways for universities and clinical labs and hospitals to work together to share samples, because for, say, blood is so hard to get for certain types of cancer patients and other types of disease. And so for people making better and better models, better technologies for liquid biopsy, like people looking at DNA sequencing and circulating tumor DNA, there's just not enough sharing of samples to be able to make big enough data sets, because there's limited number of patients for these diseases. And I think the biggest thing, which may not be something that can be immediately solved, is how do you get people to really share samples of blood from patients to be able to understand, to get them plugged into models that might never really get down to you can't just take one patient when you want to see a whole ecosystem of a disease or see all the variants of the disease. But how do we get better sharing of samples and blood and data for everybody to be able to do this work together?
That was a great answer. All right, Daniel, thank you so much for joining us today, and thanks for introducing us to nanosensing technology and your amazing work. It's been good to give students a feel for such a new and exciting field of research like yours, and to talk about collaboration and explainable AI. Thanks for taking time to join us on Pitt's Health and Explainable AI podcast.
From the University of Pittsburgh, thanks everyone for tuning in and for following the series. Join us again on HexAI, where we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine.